क्या करूँ डायरेक्ट?
आपको जैसा अच्छा लगे।
मतलब इधर ला के डेस्कटॉप में?
हाँ, कॉपी कर सकते हैं।
कॉपी नहीं, वो ठीक है। फिर इसको फुल स्क्रीन।
अभी 10 मिनट अभी सर,
हाँ
अभी रोक ले अभी।
हाँ, फुल स्क्रीन।
रिकॉर्डिंग कर देते
अभी से स्टार्ट रिकॉर्डिंग। ये बाहर कुछ जा रहा है, ये तो पंगा हो गया।
हेलो। हेलो।
अच्छा, मुझे एक बात बताएं, इसे मुझे एक चीज़ करना होगा, बहुत कुछ बाहर जा रहा है, तो अभी भी मैं एडिट कर सकता हूँ?
अभी एडिट करना क्या?
बस ज़्यादा टाइम तो नहीं लगेगा। ये तो कुछ पता ही नहीं ना कैसे क्या होता है। एंड शो, हाँ
कर।
मैं कर लेता हूँ, क्योंकि आ जाइए।
हेलो।
अच्छा, आवाज़ का कुछ कैसे पता चलेगा, मतलब वॉल्यूम का।
हेलो। हेलो।
शुरू करो। नहीं, आवाज़ को एक बार मुझे बता दीजिए। आवाज़ ठीक? हाँ, मतलब मुझे पीछे की तरफ मैं नहीं बैठ सकता।
आपका आवाज़ ज़्यादा है, स्टूडेंट लोग आपका आवाज़ ज़्यादा है।
Good morning, dear students. Are you all right?
Yes, sir.
Okay, thank you, thank you,
Yes, sir.
thank you.
Yes.
So, we are now ready to start with our job. Today's topic is the cell. Now, in your higher secondary classes, you have read a lot about the cell structure, ultrastructures, organelles, and their functions. Here we will elaborate little bit about the cell physiology, biochemistry, and some structural specializations in certain kinds of cells. The book that I will follow is called Histology, a Text and Atlas by Michael Ross and Bosley Paulina. In fact, this is the best book that we use in our department. Now, what you are supposed to read under cell is identify the different parts of an animal cell that you are already familiar. Draw a diagram of an animal cell and label the following like cell membrane, rough and smooth endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes, endosomes and microtubules. You are familiar with all those organelles and how to draw and where to draw. The third part is describe the structure and function of cell membrane, specify various pathways for exocytosis and endocytosis, and formation of lysosomes. Describe lysosome storage disease. Fourth one is describe the components of cytoskeleton, their structure and role in various physiological processes. Fifth, classify the intermediate filaments, their distribution and role in various intra and extracellular functions. Now you are all familiar with that cells are the structural and functional units of our body, and they perform a number of important functions and help in our daily activities like digestion, absorption of metabolites, removal of waste materials, sensory signal transduction, respiratory, and protection from immunity. Now these cells are diverse in their structure as well as function, and this could be seen in the specialized proteins or structures associated with them. The cell shape may vary from, say here I am showing low cuboidal epithelium to flattened cells, here large cells which are neuronal cells of ganglion, and long flattened cells with long nuclei as seen in the smooth muscle cells that line the gut. So these cells vary highly in shape and size, but in spite of that they can perform similar functions like synthesis of ATP and protein synthesis. But some cells are specialized to such an extent that they have some specialized features by which they can perform specialized functions, like although we know all cells contain little bit of contractile proteins, some cells like muscle cells, especially skeletal muscle cells, are endowed with high content of contractile proteins like actin and myosin that help them in the muscular contraction. Accordingly, their shape and size is also modified to fit into a biological structure. So, these contractile proteins, what we call useful for muscular contraction, are localized in a particular compartment of the cell called cytoplasm, where organelles are located that synthesize these contractile proteins. So, we will come to about this concept about the so-called cells containing membranous organelles and non-membranous organelles. Membranous organelles simply means those that are lined with plasma membrane like rough endoplasmic reticulum, lysosomes, mitochondria and Golgi complexes. And non-membranous organelles means which are without this lining of plasma membrane like filaments and also inclusions like lipids and pigments that are present in the cytoplasm. Now, little bit about the functions that you are familiar. I will just brush up so that you can join this lecture smoothly. Organelles, under organelles like plasma membrane involved in ion and nutrient transport, cell signaling and cell-to-cell communication. Then RER in protein synthesis, smooth endoplasmic reticulum in the detoxification of alcohols and xenobiotics, Golgi complex which is involved in post-translational modification of proteins, sorting and packaging of molecules for secretion of or transport to other organelles, mitochondria involved in ATP synthesis and apoptosis, lysosomes in digestion and autophagy, paroxysms involved in oxidative digestion of long-chain fatty acids and also detoxification of compounds like hydrogen peroxide, and cytoskeleton helping in cell motility, cell-to-cell adhesion and intracellular and extracellular transport. So here I will showing some ultrastructural features of cells and especially the organelles. Here in the first picture, upper picture, you can see these are the stacks of rough endoplasmic reticulum that synthesize proteins. These protein granules are stored in what we call secretory vesicles. These black dots are secretory vesicles. Smooth endoplasmic reticulum, smooth in the sense that they don't have ribosomes, their surface appears smooth. They are involved in synthesis of lipids and modification of glycogen, etc. This is one picture showing rough endoplasmic reticulum in the formation of vesicles, the proteins that have been synthesized by it, and in the formation of packaging into vesicles. These are Golgi complexes. You can see the Golgi have smooth surfaces, but their terminal ends are dilated. Mitochondria, here there are many mitochondria, and these are all dilated tubular cristae. Lysosomes, this is from pulmonary alveolar macrophages containing lots of lysosomal granules, small electron dense particles. These are lysosomes, and after digestion lysosomes form the so called residual body, and this residual body can also be traced to aging cells like neurons, adrenal cortex and muscle cells, where this residual body appears to contain some components called lipofuscin, which are the undigested materials of proteins and lipids that are packaged inside a lysosome, a type of residual body. Now, first important aspect is structure of plasma membrane and function. As we know that from our higher secondary class little bit, that plasma membrane is mainly composed of cholesterol, phospholipids and proteins. This, what we call the lipids, are asymmetric in the sense that they are asymmetrically distributed along the leaflets of the plasma membrane, and they are amphipathic in nature, that is, they have a hydrophilic part and that have a hydrophobic part. So they are in part hydrophilic and in part hydrophobic in nature. The phospholipid chains of these molecules, lipids, forming the inner leaflets of the plasma membrane, whereas the polar ends, the globular heads of these phospholipids, forming the outer leaflets of the plasma membrane. Many proteins are embedded in the components of the plasma membrane that are called integral membrane proteins, like here integral membrane proteins. Besides, there are many peripheral membrane proteins that remain on the outside of the plasma membranes, and these peripheral proteins may remain bound to carbohydrates forming glycoproteins, or also to the lipids forming glycolipids. So these glycolipids and glycoprotein complexes form what we call glycocalyx, present on the external surfaces of the plasma membrane. Now, in specialized regions and in localized regions of plasma membranes, we can have higher concentration of phospholipid molecules, cholesterols and specialized proteins as receptors and enzymes and substrates that collectively form what we called lipid wraps. These lipid wraps actually act as signaling platforms through which signaling interactions, signal transduction occurs more efficiently because of the closer proximity of the proteins associated with these lipid wraps. Now, regarding the integral membrane proteins, they are best visualized by a method called Fritz fracture replica of plasma membrane. So, in this method, when cells or tissues are frozen immediately in isopentane and then clipped by a cool knife at minus 190 degrees Celsius, then the plasma membrane clips along its inner leaflets, hydrophobic inner leaflets, exposing its two surfaces, one towards the exterior, which is called E phase, and one towards the cytoplasmic side or the protoplasmic side, that is called P phase. So this preparation, when seen under a transmission electron microscopy, will show the adherence of several particles which are nothing but the integral membrane proteins. Now there are many such proteins embedded into the plasma membrane of biological cells, and they can be classified broadly into 6 groups like pumps, channels, receptors, linker proteins, enzymes and structural proteins. Now movement of various substances occur via these transmembrane proteins like pumps that helps in the transport of ions along the plasma membrane. Then there are channels that helps in non-specific bidirectional movements of ions, small molecules and also water. There are receptors that can sense ligands, that is substances that they can recognize and bind to the extracellular surfaces, like is seen in the mechanism of endocytosis. Linker proteins, which are actually intracellular proteins that help in the stabilization of cytoskeleton like actin, to extracellular matrix proteins like fibronectin. There are also enzymes embedded in the plasma membrane. These enzymes have some other functions like ATPase are involved in helping in transport of ions at the cost of ATP hydrolysis. And also there are enzymes called disaccharidase and dipeptidases present on the glycocalyx of the microvilli, which are in fact the digestive enzymes digesting carbohydrates and peptides. And the 6th one is structural proteins that help to establish the intracellular connection between two cells, like seen in the polarized cells of the epithelium. Now this plasma membrane has capacity to transport molecules from outside to interior of the cells, but the question is, does plasma membrane allow everything to be transported across it? No. The answer is no, because the plasma membrane is selectively permeable. It is selectively permeable. It allows fat soluble small uncharged molecules to pass through it by simple diffusion. Means these substances can diffuse simply across the plasma membrane. Besides this, the other molecules need some proteins which are called transporters for their movement across the plasma membranes. These transporters can be divided into 2 groups like carrier proteins. Carrier proteins like small water soluble proteins that can bind to carrier proteins. After binding, there is a conformational changes in the carrier protein molecules, and then they release the substances across the plasma membrane. So this is one example of how carrier proteins utilize for transport of small water soluble proteins. So in this case, there may be involvement of energy. So when energy is involved, it is called active transport, like as we see in the transport of sodium and potassium via sodium potassium pump and hydrogen via hydrogen pumps, which are active transport, cases of active transport. On the other hand, there are molecules like glucose that can enter and passes the plasma membrane via passive transport. Now in the second category, which are called channel proteins, these channel proteins are ion selective, so they are activated only when some activity is needed in this cell. For example, in case of voltage gated ion channels which are present in the neurons, these ion channels are activated only when membrane depolarization is there in the neuronal membranes. only then these channels are open and they allow certain ions to pass through. And in the second case, there are ligand gated ion channels which are present, for example, in the neuromuscular junctions. One example is like acetylcholine receptors that are activated only when the ligand, it's their ligand, the molecule called acetylcholine binds to these ion channels and activate this channel pathway for permeabilization of certain ions and also transmitter molecules like acetylcholine. In fact, at this stage, I tell you that these receptors are nothing but ion channels, because these receptors have many forms and that collectively form a channel like structures, like you can see here many subunits that collectively form a channel like pore through which ions and substances like neurotransmitters, here I am telling about acetylcholine with ligand gated ion channels like acetylcholine receptor, can pass through the neuromuscular junction and then allows the beginning of what we call depolarization or excitatory postsynaptic potential with acetylcholine. Other substances like liquids and small and large protein molecules and bacteria and remnants of dead cells can be transported between different compartments of the cells by a mechanism called vesicular transport. So this is simply a mechanism by which different molecules are transported between different cellular compartments utilizing the plasma membrane, from where the vesicles are formed and where the vesicles collect these extraneous molecules. Now, vesicular transport can be broadly explained in terms of endocytosis and exocytosis. These two are the mechanism by which substances enter the cell and then leave these cells utilizing plasma membrane at the substrate where the vesicles are originated, and then the vesicles again fuse with the plasma membrane and again recycled to the cell for future use. Now, out of this, endocytosis has been nicely investigated by the use of transmission electron microscopy, and it has been found that this endocytosis may involve the use of certain proteins called clathrin in the formation of vesicle. So when clathrin is involved in the formation of vesicle, this is called clathrin dependent endocytosis, and if clathrin is not involved in the vesicle formation, it is called clathrin independent endocytosis. So based on this, we can have three subclasses of endocytosis, like receptor mediated endocytosis where receptors are involved, pinocytosis and phagocytosis. So first we start with receptor mediated endocytosis. As the name suggests, here receptors are involved in the internalization of substances, what we call endocytosis. So here specific receptors, as shown in the red color, these specific receptors assemble in a specialized region of the plasma membrane called lipid wraps, and then these receptors, membrane bound receptors, plasma membrane receptors, which are broadly called cargo receptors, can recognize cargo molecules, suppose here cargo proteins, for recognition and binding to their Y fragment, to their surfaces, and once this is bound to the cargo receptors, one multifunctional protein called adaptin becomes activated, and this binds with the intracellular part of the receptor, and Once adaptin binds to these receptor ligand complexes, clathrin comes into play. They assemble in the form of a coated peat. So these are the clathrin molecules. They bind to adaptin and receptor ligand complex, and this coated peat ultimately converges into a vesicular structure. Into this vesicle, these ligand receptor complexes are thrown into the lumen, and then one large GTPase called dynamine helps in the pinching of this vesicle from the apical plasma membrane, a process that is called vesicle budding. After this, once this clathrin coated vesicle moves to the interior of the cytoplasm, this coat is released, that is, clathrin molecules are released, they are recycled for future use, and the uncoated vesicle, this uncoated vesicle is now ready to fuse with other organelles that we will deal now. So what will happen after this? After the uncoated vesicles are formed, these uncoated vesicles, suppose these clathrin molecules are now released, shed, and the uncoated vesicles will fuse with an organelle called early endosome. This early endosome lies close towards the periphery of the cell and adjacent to the plasma membrane, almost adjacent to the plasma membrane, and it has a tubulovesicular structure. It has a tubular part and vesicular part. This early endosome is involved in sorting of the materials that has been internalized. So it has two parts, one is ligand and the receptor. So all receptor components are transported or sorted to a structure, tubular structure of this early endosome, and they are then recycled to the plasma membrane for future use. The ligands, the substances that are internalized, may be protein molecules, these are sorted out, these red dots, these are then sorted out once they are clipped by the acidic pH of these early endosomes. From these receptors, these protein molecules, suppose these protein molecules are concentrated in the vacuolar structures, and ultimately they are separated in the form of a structure called multivesicular body. This multivesicular bodies then fuse with another organelle called late endosome. In the late endosomes, these materials are delivered. Here the pH is 5.5. Late endosomes, early endosomes have pH of 6.2, and late endosomes have a pH of 5.5. Increased acidification goes on because of the opening of this hydrogen pump that allows hydrogen to permeate from the cytoplasm, and with increased acidification, when the pH is lowered to 4.5 to 4.7, these late endosomes eventually mature into lysosome. If we see a difference between the early and late endosomes, we can see that these early endosomes are towards the periphery of the cell, whether late endosomes are very close to the Golgi complexes. The pH of early endosomes is 6.2 and that of late endosomes is 5.5. Late endosomes are typically tubulovesicular structures, whereas late endosomes are vesicular in shape. The function of early endosomes is to sort out the endocytose proteins, receptors and ligands, whereas the late endosomes receive multivesicular bodies, as I told you, multivesicular bodies packaged with proteins and other substances. And these late endosomes also receive transport vesicles that are budded up from the trans-Golgi network, along with the acid hydrolysases, that is the lysosomal enzymes. So these late endosomes ultimately transform into lysosomes and then they mature. So you have to know about the differences between early and late endosomes and their function. Another process of endocytosis is pinocytosis, by which liquids and small protein molecules are internalized into vesicles pinosomes called pinosomes. Here clathrin is not involved in the formation of these pinosomes, so this is called clathrin independent endocytosis. And so the vesicles have a smooth surface, unlike your receptor mediated endocytosis where the vesicles have a clathrin coat. So here the vesicles are smooth and their size is also small, like these vesicles are smaller than 150 nanometer in diameter, contrary to the vesicles seen in receptor mediated endocytosis, which are somewhat larger than 250 nanometer in diameter. Also we can see there are bud-like structures that originate from plasma membrane, which are called caveoli. So these caveoli are much larger, even they are smaller than 50 nanometer. They also help in the transport of certain substances like cholesterol, because they have receptors, cholesterol binding domains on their membrane, so they can transport cholesterol into the cytoplasm. So these caveoli are also structures that help in pinocytosis. The third is phagocytosis. This phagocytosis is an actin dependent endocytosis. So what happens in phagocytosis when bacteria or other foreign bodies enter our system? The humoral immune system is activated and this leads to the formation of antibodies. These antibodies then bind to the surface of these inverted pathogens like bacteria. These antibodies have two structures, one this kind of arms and one this type of tail, which is called Fc fragment of antibody. These two are called FAB fragment. So this Fc fragment remains open and this Fc fragments are recognized by Fc receptors present on the plasma membrane of professional phagocytes like monocytes, macrophages, neutrophils, dendritic cells, osteoclasts, which can recognize this kind of substances. Then there is a formation of pseudopodia into those professional phagocytes by throwing of the cytoplasm. In this case, there is reorganization and polymerization of actin molecules in the formation of or throwing of pseudopodia. So this is why it is called actin dependent endocytosis. The pseudopodia then engulfs this bacteria and these bacteria are then localized in a large vacuolar structure which is called phagosome. This phagosome is then internalized, and once this phagosome start to travel to the interior of the cytoplasm, they receive the transport vesicles containing the hydrolytic enzymes, newly synthesized hydrolytic enzymes, lysosomal enzymes. So they will fuse these hydrolytic enzyme bags will fuse with this phagosome and ultimately this phagosome will convert into late endosomes and into lysosome, and then this lysosomal hydrolysis will digest the materials, maybe bacteria or some other substances. Here you can see in the formation of phagosome there is no involvement of clathrine, that is the phagosomes are not lined with clathrine, but here actin is involved. So this is called clathrine independent, actin dependent endocytosis. In the same way, non-biological materials like inhaled carbon particles, dust and cell debris are internalized and they are then digested via formation of phagosomes. But here the receptors are non-specific, means a number of multiple receptors can participate in the engulfment of the substances. This only depends on the structures that are in the close contact of those receptors and their shape and size. So this is somewhat non-specific, but it is always specific, wherein the Fc receptors are involved for this engulfment of antibody, a process sometimes called opsonization. So we have read about endocytosis. There are three types of endocytosis, subtypes: receptor mediated, pinocytosis and phagocytosis. Another method of vesicular transport is exocytosis, means by which the vesicles once formed inside the cytoplasm then travel to different compartments of the cell and then they are transported and they are transported to the apical plasma membrane, where these vesicles fuse with the apical plasma membrane to release the content outside the cell, a process called exocytosis. So this begins with the formation of substances like proteins in the cisternae of rough endoplasmic reticulum. So these are the stacks of rough endoplasmic reticulum where ribosomes synthesize the proteins. These are the ribosomes, and these ribosomes synthesize proteins, and these proteins are then packaged into vesicles, means these proteins are then separated out in the vesicles which are called transport vesicles. Now this vesicle will now start moving inside the cytoplasm. So from the rough endoplasmic reticulum, they will start migrating towards the cis phase of the Golgi network. This is the entire Golgi network, and the part lying closer to the RER is called cis Golgi network. So these vesicles will move and reach to the cis phase of this Golgi network, and there they will fuse. Ultimately these proteins will be transported via this cisternae of Golgi complexes, and finally they will reach to the trans Golgi network, which is located just opposite to the cis phase of this Golgi complexes. So here in the trans Golgi network, these vesicles are formed via the separation of these proteins into these vesicles, and these vesicles are now called secretory vesicles because they are ready to be secreted outside the cell. So these secretory vesicles will move towards the apical cytoplasm, and then they will fuse with the apical plasma membrane to release their content outside the cell. So this is one mechanism of vesicular transport by which synthesized materials are transported outside the cell, as happens in the secretory cells like plasma cells or by fibroblast secreting proteins outside the cell. And once this happens, the remnants of these vesicular membranes are internalized by endocytosis and they are again recycled. This exocytosis has two pathways by which the molecules can leave the cell. One is called constitutive secretory pathway another one is regulated secretory pathway. In the first, that is constitutive secretory pathway, the molecules once packaged in the secretory vesicles reach the apical plasma membrane and then they are released outside the cell. So this is constitutive, means it is an ongoing process. It happens continuously inside the cell without the presence of any trigger or stimulus. So this is called constitutive secretory pathway, as seen in the release of antibodies by the plasma cells and pro-collagen molecules by the fibroblasts. There is another method that is called regulated pathway of exocytosis, in which secretory molecules are released outside the cell only in presence of certain stimulus. Here, under presence of certain stimulus, say when calcium influx happens inside the cell via calcium channels, there is an increase in the calcium level in the cytosol, and this calcium ions then bind to the vesicular membrane, ultimately helping them in docking with the plasma membrane, apical plasma membrane, and their fusion with the apical plasma membrane, thereby helping in the release of the content outside the cell. So this is called regulated secretory pathway of exocytosis. So this happens in structures like pancreatic exocrine acinar cells that needs some stimulus for the release of exocrine materials like enzymes, and also in the nervous systems release of neurotransmitters. So there are two pathways. Now there are certain proteins which help in the vesicular transport from rough endoplasmic reticulum to Golgi complexes, and also from Golgi complexes to rough endoplasmic reticulum in occasional cases. So these proteins are in general called coatomers or coat proteins. Coat proteins which help in the vesicular transport between RER and Golgi complexes and back. One such protein is called COP2, that is coat protein 2, that is involved in the anterogate transport. Anterogate transport means transport from the RER to the cis phase of the Golgi network. So here you can see COP2 coated vesicles are formed from the rough endoplasmic reticulum, and then they fuse with the cis phase of the cis Golgi network. Then this vesicle travels and then they reach to the trans Golgi network. Now in this process it may happen that sometimes by mistake misfolded proteins can be transported in this along this pathway. Golgi can recognize all those misfolded proteins and they are again sent back to the rough endoplasmic reticulum via COP2 coated vesicles. So these are COP1 coated vesicles which are involved in transporting these vesicles back to the rough endoplasmic reticulum, a process called retrograde transport from Golgi to RER, whereas RER to Golgi is called anterogate transport. So COP1 and COP2 are involved in retrograde and anterograde transport of vesicular materials respectively. Here you can see that even among the all other cisternae of Golgi complexes, this transport happen, and this is mediated by COP2 from here to here. So COP2 is involved, but from trans Golgi network to RER, this is always mediated by COP1. So this is to be remembered. So one part, biogenesis of lysosomes, is very important in your curriculum. Although you will read a lot under biochemistry, and tomorrow maybe you have one class mm, mm, for this kind of organelles and structures and functions like plasma membrane, where you will read in detail about the structure of plasma membrane, their functions, etc. Theories involved with plasma membrane like fluid mosaic model describing how integral membrane proteins move along the plane of the plasma membrane, meaning of fluidity of plasma membrane, and the proteins and phospholipids involved in fluidity, etc., etc. Formation of lysosomes. You will read in biochemistry and to some extent also in the physiology classes. So at this stage, you don't have to be very worried about this lecture content, but you just read and you just brush up and you integrate your knowledge from physiology and biochemistry, where the faculties are ready to discuss in detail everything about these organelles, their structures and functions. So here we will discuss about the biogenesis of lysosomes, how lysosomes are formed in a cell. You know from your higher secondary background that lysosomes possess hydrolytic enzymes which are called hydrolases. Initially they are pro-hydrolases, means inactive enzymes. Besides these digestive enzymes called acid hydrolases, lysosomes also contain many important proteins in the form of lysosomal membrane proteins that could be classified into three: lysosome associated membrane proteins, LAMPs, lysosomal membrane glycoproteins, LGPs, and lysosomal integral membrane proteins, LIMPs. So lysosomes contain all these proteins besides hydrogen into their lumen. We know that although these enzymes are active in digestion of materials that are internalized inside the lysosomes, the lysosomal membrane itself is resistant to digestion by these hydrolytic enzymes. This is because of two reasons. Number, these lysosomal enzymes are heavily glycosylated, that is, sugar residues are added to these enzyme protein molecules, and these sugar residues are constantly facing the enzymes. So the protein part is resistant to digestion by this hydrolysis. Besides, there is one unique phospholipid called lysobisphosphatidic acid that also helps in resisting to digestion of the lysosomal membrane proteins by lysosomal enzymes. So there are still controversies about the biogenesis of lysosomes. Many authorities do not believe various concepts, existing concepts, etc. But what we have learned from textbooks and what the most authorities suggest and believe is that what I will talk here. Now here accordingly two concepts are to be noted. Number one concept, how the hydrolytic enzymes are synthesized, and number two concept, how the lysosomal membrane proteins are synthesized. Now in the first, formation of hydrolysis, you can assume that this process, hydrolysis, which are proteins, so they will be synthesized in the cisternae of rough endoplasmic reticulum. But during their formation and modification, these enzymes proteins are modified in such a way that they hold a capacity to add certain molecules called mannose 6 phosphate. This mannose 6 phosphate acts as a signal or as a tracer, means lysosomal enzymes. all lysosomal enzymes will contain this mannose 6 phosphate, and they are recognized, all lysosomal enzymes with this mannose 6 phosphate residues will be recognized by its receptor called mannose 6 phosphate receptors, which are present in the trans-Golgi network, early and late endosomes. Okay. So once synthesized, the prohydrolases modify in such a way that they can hold this mannose 6 phosphate, and then these enzymes are packaged into the transport vesicles, clathrin coated transport vesicles by trans-Golgi network. From the trans-Golgi network, these vesicles will start moving towards the early endosomes where the receptor for mannose 6 phosphate are located. So this mannose 6 phosphate will bind, and the enzyme complexes will be internalized inside the lumen of early endosome. This early endosome will be eventually transformed into late endosomes, where all these molecules will be released in presence of an acidic environment. This receptors in fact remains bound to the membrane of this late endosomes. Then this late endosome will ultimately mature into lysosome. So this is one mechanism by which the lysosomal hydrolases are synthesized and transported to early and late endosomes, and then finally converted into lysosomes. The other component of the lysosome is lysosomal membrane proteins. So they are also synthesized in the cystine of rough endoplasmic reticulum, but they do not bear this kind of tag like mannose 6 phosphate. So automatically they are targeted by different mechanisms. So in one mechanism, that when the lysosomal integral membrane proteins are synthesized, they are packaged into transport vesicles, they pinch off from this trans-Golgi network, and immediately they are released to the outside of the cell by exocytosis. So this is one process by which the lysosomal integral membrane proteins reach outside the cell via exocytosis. After this, the same molecules which are now released outside the cell will be engulfed and endocytosed in clathrin coated vesicles. So this vesicle will now contain all lysosomal integral membrane proteins, and this vesicle will be transported to early endosomes where these integral membrane proteins will be released. And in another mechanism, the Golgi sequester the protein molecules like lysosomal membrane glycoproteins and lysosome associated membrane proteins, and in coated vesicles they will be directed straight to the early endosomes and to late endosomes. Both might happen, it depends whichever is present in greater amount. So both early and late endosomes are involved in capturing this kind of vesicles, coated vesicles that contain lysosomal membrane glycoproteins and lysosome associated membrane proteins. So they will be delivered to early and late endosomes. Now these enzymes are recognized by Golgi complexes, proteins. These proteins are recognized once synthesized by ARIA. These proteins are recognized by Golgi cystine by one unique sequence cytoplasmic short terminus that can recognize adaptin. Now once yes sir.
So which sequence?
One short cytoplasmic sequence that has affinity towards adaptin. Now, once adaptin binds to those membrane glycoproteins, then adaptin has an also affinity towards clathrin molecules, so clathrin coated vesicles will be formed. This is how the protein molecules as membrane glycoproteins are separated out from the enzyme molecules which are tagged to mannose 6 phosphate. So these materials, late endosomes, will ultimately convert into lysosome. So this is the most accepted view of biogenesis of lysosomes. Although there are other theories available, we do not know actually, but this is the most accepted theory of biogenesis of lysosome. So this is what is also written in another book called The Cell, a Molecular Approach by G.M. Cooper 2000. There also it is said that these transport vesicles once formed with acid hydrolases, they will fuse with late endosomes, and these late endosomes via increased acidification, because proton pumps remain open, via increased acidification will release these enzymes and protein molecules, and ultimately these structures will convert into lysosomes. So one function of lysosome is to digest the materials that arrive to the cell via endocytosis. Now these are extracellular, but there are also cases where intracellular particles and dysfunctional proteins can also be needed to be digested by lysosomes, and here lysosomes help in the intracellular digestion of dysfunctional organelles and also protein particles by a mechanism called autophagy. It was termed by Christian D. Dubé, who also discovered lysosomes long back in 1963 from Belgium, Christian D. Dubé, who got Nobel Prize. Now 2016 Nobel Prize also delivered to one Japanese group, Yoshinori Osumi, for showing the genes involved in autophagy. These genes are called autophagy related genes, or ATG in short. So what happens in case of autophagy? First, there is an induction of isolation membrane from smooth endoplasmic reticulum, and this isolation membrane will then engulf the dysfunctional organelles like mitochondria or lysosomes, and then or ribosomes or other organelles. Then this membrane surround this structure, forming what we call autophagosome. This autophagosome will then fuse with lysosome, forming autolysosome and digesting the dysfunctional organelle or misfolded proteins, etc., by a process called autophagy. Now, when large organelles are involved in autophagy, this is called macroautophagy, like mitophagy, that is autophagy of mitochondria, and microautophagy, where small protein molecules are involved, called microautophagy. Now a little bit about lysosomal storage disorder. These disorders happen due to enzyme deficiency, means when we acquire some mutations in the genes that encode those lysosomal enzymes. As a result, lysosomes cannot break down certain components, and they start on piling up inside the lysosomes, forming disease phenotypes like Butcher's disease, in which there is a deficiency of glucose erythrosidase, and the accumulated product is ceramide, which is toxic to the cell, something like that. So you read little bit about this lysosome storage disorder. Next is cytoskeleton. Cytoskeleton is a network of filaments and microtubules. Among the filaments, we have actin filaments and intermediate filaments. The function of cytoskeleton, this is always plural, function of cytoskeleton is to provide resistance to cell deformation in cell migration and elongation as helped by actin, in phagocytosis by actin, segregation of chromosomes in cell division by microtubules, intracellular transport by microtubules, and muscle contraction by actin together with myosin. See little bit about the comparison of this cytoskeletal element. Like actins have a diameter of 6 to 7 nanometer, intermediate filaments 10 nanometer, and microtubules 25 nanometer. Actin filaments are polar, as is the case with microtubules, but these intermediate filaments are non-polar, so they are stable. These filaments can elongate and shorten, likewise the microtubules that can polymerize and depolymerize by a process called dynamic instability. That is, microtubules keep on elongating and shortening by this process called dynamic instability to keep in contact with the cell periphery. Actins occur in bundles, intermediate either in bundles or separately, whereas microtubules always occur separately. Their functions I have already told. So cytoskeleton can also form apical domain of the cells like cilia and flagella and phylopodia, lamellipodia and macrovilli that collectively form the apical domain cytoskeleton. Cilia, flagella, yes?
Excuse me, sir. Sir, in the previous slide, what was the meaning of actin filaments are polar?
They can polymerize. They can polymerize with their monomers.
The ability to polymerize is called polar.
Yes, yes, like
Okay, sir. Thank you.
like microtubules, they are formed of alpha and beta tubulin, and this beta tubulin can bind to GTP, and then they can polymerize. So that is why it is called polymerization.
Fine, sir.
Okay, thank you. So in
Thank you,
cytos,
sir.
okay. So in cilia, flagella, you can see structures called microtubules. So these are microtubule based structures. Whereas microvilli and phylopodia, lamellipodia, these are actin based structures. So this is one transverse section of cilia, ciliary appendage called axoneme, where you can see the microtubules. And when this is cut in cross section, you can see this kind of structure with doublets of microtubules. There are 9 doublets arranged peripherally and one central doublet. Okay, so this arrangement is called 9+2 configuration of microtubule doublets formed of microtubule A and B that are connected by certain structures called radial spoke to this central unit. Now these microtubules remain united to certain proteins called dynein, outer dynein and inner dynein arms, and there are problems where these dynein molecules disorganize and sometimes leading to the disappearance of these peripheral doublets or even central doublets, collectively forming a pathological sequence called ciliopathy, like in primary ciliary dyskinesia, where these molecules may disappear, creating what we call immotile cilia syndromes. That is, the cilia cannot move, ciliary beat stops, as happens in the respiratory and nasal epithelium. So if we compare the apical domain cytoskeleton, you can see microvilli have a length of 1 to 3 micrometer. Then cilia are differentiated into primary cilia, which are non-motile because they don't have the central doublet. So this arrangement is called 9+0 pattern. So these are called primary cilia. Their length is somewhat longer, 2 to 3, And there are motile cilia which are much longer, 5 to 10 micrometer. These motile cilia are composed of axonyms with microtubules having 9 plus 2 doublets along with certain motor proteins that help in their movement. So these are motile cilia and primary cilia. These primary cilia are present in all cells and their number may vary from 3 to 5, while motile cilia number is variable and they are present in structures like olfactory epithelium, trachea, bronchi, oviducts, palms and ependyma. Another type of structures is there called stereocilia, which are long structures 80 to 100 micrometer, long 80 to 100 micrometer, which are actin based structures like microvilli. Primary cilia and motile cilia are microtubule based, but microvilli and stereocilia are actin based apical domain cytoskeleton, and they are capable of regeneration like as we see in the cells of epididymis and in hair cells of inner ear. So both microvilli and stereocilia are absorptive and they also show passive movement, whereas these primary cilia are mainly mechanoreceptors, that is they transmit signals from extracellular environment to the cell, and motile cilia mainly function to transport secretions and foreign bodies on the cell surfaces. So this is one picture showing the brush border of intestinal epithelial cells which are composed of microvilli. This is one electron micrograph showing the microvilli having actin core. You can see the actin filaments. These are the stereocilia, long cilia, and these are the structures called microtubules which appear as isolated structures involved in intracellular transport. Now this cytoskeleton, actin and intermediate filaments also help in the formation of junctional complex, like actin is involved in junctional complex like zonula occludens, that is tight junctions, also in zonula adherens in intermediate junctions, zonal adherens in the intermediate junctions between epithelial cells, and intermediate filaments that are involved in the formation of junctional complexes in the form of macula adherens or desmosomes that are formed between two epithelial cell surfaces via interaction with some proteins, scaffold proteins like desmoglein and desmocollin, which remain bound to these intermediate filaments. You will read details about these cytoskeletal proteins and their involvement in junctional specialization when you read the epithelial tissues. So there are 6 types of intermediate filaments: type 1 and type 2, acidic and basic keratins like what we see in epithelial cells, which are called epithelial keratins or cytokeratin, and in hairs called hair keratins. There is type 3 intermediate filament called in various terminologies like desmin, present in skeletal muscle, glial fibrillary acidic protein present in astrocytes of the nervous system, peripherin in the peripheral neurons, vimentin present in fibroblast and endothelial cells. Type 4 intermediate filaments are characterized by their presence in the axons as neurofilaments and in muscles as synamin and syncoilin. So these are all type 4 intermediate filaments. Type 5 intermediate filament contains like nuclear lamins that line the nuclear envelopes and helping in providing architectural stabilization of the nuclear envelope. Lamins, intermediate filaments. Type 6 is nestin, an intermediate filament protein that is present in the early neuronal precursor cells. So these are electron micrographs. One electron micrograph showing the glial fibrillary acidic protein rich filaments, intermediate filaments in an astrocyte. This is showing the presence of neurofilaments, these black dots, and few microtubules in the axonal compartments. So these neurofilaments are examples of type 4 intermediate filaments. So the theory part is over, and now I will reach to the practical demo aspect of the lecture: cells, preparations and staining methods that you are supposed to see in the demonstration classes. Anyway, it is not a major issue. You can understand from this presentation also. So first aspect is how tissues are prepared for histological observation under a light microscope, and the basic staining method by which we can differentiate the components in a cell or tissue. So the aim of histology is to see the microanatomy of cells and tissues and organs, and to correlate the structures that we see with their function. So tool can be light microscopy. In the beginning, light microscopy is just good enough to see the histological organization of cells and tissues. But in certain cases, when we need detailed interpretation of microanatomy like ultrastructure, then we can see the preparation by a different method to see under electron microscopes like transmission and scanning electron microscopes, because they afford higher resolution than light microscopy, and their final details of organelles or other structures can be seen nicely. Besides light microscopic observation of tissue structures, sometimes it may be essential to see the chemical organization or the specific chemical moieties present in a cell or tissue by a method called histochemistry. And in many cases we need to show, we need to see the specific molecules like neurotransmitters, neurochemicals present in the nervous system and the gut enteric neurons by a method called immunocytochemistry, where we utilize antibodies to bind to the target particles like proteins or neurochemicals, wherein these chemicals act as antigen. So this technique is based as antigen antibody reaction to see the localization of protein particles, neurotransmitters, receptors, etc. in the nervous system and in many other tissues. And the third one is in situ hybridization, whereby we can localize the synthesis and formation and fate of nucleic acids like DNA and RNA, their formation, rate of formation, their degradation and their movement, like movement of mRNA, etc., by a method called in situ hybridization. Now here we will mainly focus with tissue preparation for light microscopy. Now from your basic knowledge you can understand that shortly after death tissue autolysis begins. So this is called post-mortem, that is after death. This post-mortem death or post-mortem autolysis is very rapid, fast, and it is almost unavailable wherever the victim is there, in hot climate or cold climate or in Antarctica region. Post-mortem autolysis is inevitable. This is because of the triggering of the lysosomal enzymes via rupture of their lysosomal membranes that start digesting the cellular components. So in order to prevent this autolysis from undergoing autolysis, if we can somehow fix or protect these cells and tissues from undergoing autolysis, the tissue structures will be retained. So fixation is one method by which we can retain the tissue and cell structures. So this process, what we call fixation, actually preserves permanently the tissue components, and the tissue components are proteins, carbohydrates and lipids that degrade faster immediately after death by post-mortem autolysis. So if we perform fixation, this process will permanently preserve and protect the cellular components from undergoing decay. So this fixation is actually aimed to prevent enzymatic degradation of cells and tissue components by autolysis. So how to do this fixation? The chemicals that are used in fixation are called fixatives, and one classic fixative is formalin, that is 37% aqueous solution of formaldehyde, and it is always prepared as 10% neutral buffered formalin. That is, buffer that we use here is phosphate buffer, and neutral means at a neutral pH, that is 7, that this formalin is dissolved in buffer having a pH of 7. So in 10% neutral buffered formalin we can fix our desired tissues for histological preparation and in order to see under a light microscope. So what is the chemistry of this fixative? This compound formalin actually reacts with the amino groups of proteins and thereby stabilizes the protein components of the cells. And if the proteins are nicely retained, other two constituents, carbohydrates and lipids, are also retained to some extent. But in course of tissue processing, they are also lost. That I will tell later on. Another fixative is what we call Bouin's solution, that contains picric acid 25 ml, formalin 75 ml and glacial acetic acid 5 ml. So totaling 105 ml. So remember, so this is not 100, this is 105 ml containing picric acid and formalin. Both are protein fixative, and glacial acetic acid that fixes lipids to some extent, if not entirely. Now after fixation is performed, which may be continued for 1 to 2 days depending on the tissue size, generally we take a small size of tissue 3 to 4 millimeter in size. We perform a step called dehydration, means we need to remove the water content in that sample, because our ultimate target is to see that tissue under a light microscope, and tissue as such does not help us in seeing anything. But if the tissue is cut into thin slices and stain them suitably, so many details of the cellular structures can be seen. So in that process we need to dehydrate the samples, we need to remove the water content of the samples after fixation, and then we embed the samples in suitable medium that will allow section cutting, thin sectionings. Okay, so after fixation we perform dehydration. So this is generally performed in ascending grades of ethyl alcohol, starting from 50% ethanol, then 70%, 80%, 90%, and finally in absolute alcohol. So all these grades will ultimately remove the water content from the tissue. So tissue will become dehydrated. Okay. But the bad effect of this dehydration is that many tissue components like carbohydrates and lipids are lost during fixation and also in dehydration. In fixation, in formalin, will lead to loss of both carbohydrates and lipids to some extent, And if we use aqueous fixative like neutral vapor formalin, also some carbohydrates like glycogen will be leached out from the cells. And during dehydration in ethanol, major components of the lipids from the cells, cell membrane and organelles will be leached out. So these are the drawbacks when we use fixative like aqueous fixatives or formalin, or when you use ethanol as a dehydrating agent, that we lose many cellular components, especially carbohydrates, minerals and lipids. But definitely, if our target is to see carbohydrates or lipids, we can specifically fix the tissue with carbohydrate preserving fixative or lipid sparing fixatives. In that case, there will be no such problem, and as such, the exact cellular localization of lipids and carbohydrates can be seen in a histological tissue. So anyway, our main target is to see the histological organization using special stains and seeing under light microscopy. So we need not bother here. So after this dehydration, these tissue samples are embedded in molten paraffin that has a temperature of 60 to 62 degrees Celsius. So in this paraffin bath, tissues are embedded, then they are hardened by cooling, and the whole stuff is then sectioned by a device called rotary microtome. So this is one rotary microtome where the blade is kept, and this is the sample in yellow color because this tissue sample was fixed in Buin's fluid that imparts a yellowish color to the tissue, and the surrounding element is the paraffin. So this lady, maybe one technician or student or researcher or clinician or histopathologist, is sectioning from this paraffin embedded tissue, and this is called paraffin ribbon that contains these sections. You can see it. So this is one method by which we can cut sections of 3 to 5 micrometer thick, and there are other methods also by which we can cut also thinner sections, like if we don't embed in paraffin, but if we prefer using other chemicals like resins, we can cut much thinner sections, 1 micrometer or sometimes less than 1 micrometer, 0.5 micrometer. They are very thin sections, and these thin sections are cut with a different microtome which is called ultra microtome. The designing and principle is same like this, but that ultra microtome allows very thin sectioning, 0.5 to 1 micrometer, which the rotary microtome cannot afford because of the use of paraffin. This is the limitation of paraffin substances that doesn't allow thin sectioning. So anyway, our target is light microscopy, so we can use paraffin for cutting 3 to 5 micrometer sections, which are just good enough to allow light to pass through these thick sections after staining. Okay, section thickness has a matter because light should pass nicely through these thick sections so as to allow visualization of the tissue components. 3 to 5 micron thick sections are good enough, where resolution is afforded nicely to see the cellular components. Now, before staining, we must know about the chemical basis of staining, which is important for many aspects, not only from exam point of view. We must know the chemical basis of staining. Histological sections for light microscopy are stained with acidic dyes and basic dyes. So both are used, acidic dyes as well as basic dyes, and this dye will stain different components of these cells. So what an acidic dye is and what a basic dye is, we must know this. An acidic dye means one that contains a net negative charge in its coloring material. So it is represented by sodium plus and dye negative. So acidic dye carries a net negative charge. Example is eosin, whereas the basic dye carries a positive charge on its dye molecules, and it is represented by a generalized formula of dye plus chloride minus. Example is hematoxylin. And from this you can easily understand that acidic dye that bears a negative charge will definitely react with cationic groups of the cytoplasm or cells. Cationic means positive, positively charged elements inside the cell. Whereas basic dye that contains or that carries a positive charge in its dye molecules will definitely interact with the anionic components, which are negative components of the cell. So what are those? See a little bit about this. Like acidic dyes react with cationic groups, positive groups in cells, and these are present in the cytoplasm like amino groups of proteins, cytoplasmic proteins. And this affinity of this cationic groups substances like amino groups of proteins to react to an acidic dye is called acidophilia. Like cytoplasmic proteins with their amino groups can bind to an acidic dye, and so these cytoplasmic proteins are called acidophilic. Acidophilic. On the other hand, the basic dyes, there are basic dyes that react with anionic components, that is the negative component of cells, which could be phosphate groups of DNA and RNA and also proteins, and sulfate groups of glycosaminoglycans present in extracellular matrix, and carboxyl groups of proteins. So these functional groups will bind with a basic dye, and their affinity towards this basic dye is called basophilia. That is a phenomenon of the ability of these anionic groups to react with a basic dye. It's called basophilia. Like the nucleus with its DNA and RNA content is basophilic. So we simply called the nucleus is basophilic and the cytoplasmic is acidophilic, because nucleus stains with basic dye and cytoplasm stains with acidic dyes, so that you can see a differential picture of the cells. Some part is colored in a different manner which is not present in the other compartment of the cell.
Sir,
Yes, yes,
sir, even proteins have COH group in them, and that is stained by basic dyes.
basic dyes, yes. Only amino groups are reacted by acidic dyes.
So cytoplasmic proteins can be both acidic or basic,
Yes,
because
yes,
they have NH2 and COH both.
exactly, you are right. And that is why to some extent you will see that in most cases the cytoplasm is not perfectly in the color of acidic dyes. But nucleus is always basophilic, means it will impart one blue color. Okay, so this is because of the differential reaction with the cytoplasmic proteins having amino groups or carboxyl groups. Now another method or phenomenon called metachromasia is to be understood. So this metachromasia means it is a property of basic dyes basic dyes that have affinity with anionic components of cells. So what happens in metachromasia if we use basic dyes, say for example toluidine blue, then if we use very concentrated solution of these dye molecules, they will form polymeric aggregates. Because this concentrated solution of these toluidine blue molecules have affinity with ionized groups of the cells and tissues like sulfates and phosphates groups present in specialized cells and tissues. So when we use toluidine blue, which will react then ionized sulfate and phosphate groups present in cells like the ground substances of cartilage and mast cell granules which contain heparin. So the color will not appear in blue as such, but the color after absorbance by these sulfate and phosphate groups will impart a different color which may be purple to red. So this is one method by which we can study cells and tissue in differential color. That is, one part may appear purple and one in red, so that we can discriminate which part contains what. Although we cannot identify chemically, but at least we can identify the structural differentiation. Some parts stain in purple color and some part stain in red color. So this is one light microscopic picture of mast cell. Here you can see some granules stained in light purple color, and when you see these mast cells under a transmission electron microscope, these granules appear like this. These are secretory granules which are stored in the cytoplasm of these cells, and they appear as electron dense. So these granules are actually what we see under light microscope as purple colored structures in mast cells. Now once the stains are ready for histological slide staining, we can try with staining. Now as I told you in the beginning, ideally speaking, both acidic and basic dyes are to be used to see the entire components of a cell or tissue. Suppose if we use only hematoxylin, you can see nicely the nuclear parts, because nucleus or nuclei are basophilic and hematoxylin is a basic dye. So you cannot see the cytoplasmic components. On the other hand, if we see one slide stained with eosin only, we can see only the cytoplasmic part, but here no nuclear compartments are seen. So this is a drawback of staining with a single dye like eosin or hematoxylin. So we use this combined use of hematoxylin and eosin, basic dye and acidic dye for our routine histological demonstration of biological tissue sections. Like here, this slide or tissue section is stained with hematoxylin as well as eosin. This pink coloration is this due to the presence of cytoplasmic proteins in these specialized groups of cells. So other than this nucleus and cytoplasm, which are basophilic and acidophilic respectively, cytoplasmic filaments can be stained with a acidophilic dye, like in muscle cells that contain actin filaments and myosin. These filaments can be stained with an acidic dye. So those muscle cell filaments are acidophilic, and also membranous compartments that can be stained with an acidic dye, and extracellular fibrillar materials like collagens that can be stained with an acidic dye. So extracellular fibers like collagens are all acidophilic and they can be stained with an acidic dye. So this eosin and hematoxylin not only show the nucleus and the cytoplasmic proteins, but also they show the fine filaments present in the cytoplasm, intracellular membranous compartments that are stained nicely in pink color, and extracellular fibers that are present along with the cell periphery and in the intercellular spaces. So there is wide application of this basic staining to see the general histology of most tissue types. Now see the cell types. Our body contains a diverse kind of cell types and they have different kinds of functions because they contain specialized proteins in individual cell types. Not a particular cell type is in parallel with other cell type. Like epithelial tissue, the proteins present in epithelial tissue may not be present in connective tissue. Similarly, the connective tissue proteins may not be present in muscular or nervous tissue or in the vascular tissue. So there is a diverse kind of cell types present in the body and we need to see them by different kinds of staining. Although this is not the right time to learn everything, in course of time you will read everything when we will have the full course of the tissue types in our lectures. You will come to know the different staining methods to stain the different cell types and tissue components of our body. So in general, for the sake of today's discussion, we can tell that we may know that the cells of epithelial tissues can be stained with these basic dyes, hematoxylin and eosin, and they shows everything. There is no problem with this. Connective tissue that contains fibroblast cells, plasma cells, macrophages, osteocytes, adipocytes, and fibrous materials like tendons and ligaments. They can be stained with a suitable staining method called Mason's trichrome, having different dyes, at least 3. So that is why it is called trichrome, and it stains different components of the connective tissue. Okay, so cells wherever present are stained in a color which is not imparted in the fibrillar or fibrous tissue elements of this connective tissue. So this is one classic staining method, Mason's trichrome, by which we can stain the connective tissue cells and also the fibrillar materials that constitute the connective tissue. Muscular tissue in general can be stained with eosin and hematoxylin, and we can see nicely all components of muscular tissue. Although we can apply special stains, and you will come to know when you go through this chapter of muscular tissue in your lecture. Nervous tissue, which comprises neurons and glia, similarly can be stained with hematoxylin and eosin, but since many features of neurons and glia cannot be seen by this staining method with eosin and hematoxylin, we apply special stains like thionin and crystal violet, which are called Nissl stain, that stain neurons, and another stain called Golgi stain, discovered by Camillo Golgi, the discoverer of Golgi complexes from Italy. So this Golgi stain consists of silver impregnation method, like in which silver nitrate and potassium dichromate and sometimes osmium tetroxide is utilized to stain the neurons. And finally, the vascular tissue components like endothelium, pericytes, and smooth muscle lining of the arterials, arteries and veins can be stained with hematoxylin and eosin, and also with special stains that you will come to know after you reach to the stage of reading vascular tissue or conducting tissue of the body. So here I will show some hematoxylin using staining preparation just to show whether they really tell us anything about the histological organization of tissue. Like here, hematoxylin and eosin stained section showing pseudostratified epithelium of trachea. Here the nuclear components of these cells are basophilic and their protein components are acidophilic. So this red color actually indicates the protein components of these cells. And outside this cell you can see some projections which are actually cilia. So this is actually from pseudostratified ciliated epithelium of trachea. Okay. Now you can ask me why these cilia are stained in red. This is due to the fact that cilia are composed of macrotubules, and macrotubules have many proteins. Macrotubule associated proteins, tau, and many other proteins that gives this red coloration to this cytoskeleton as cilia. This is from the cerebellum, cerebellar cortex, where you can see two compartments, one stained with basic dye like hematoxylin, by which you can see very small nuclear phenotypes which are the granule cells of cerebellum, and large cells that are stained with eosin. These are in fact the Purkinje cells having lots of proteins, so they are stained with acidic dye like eosin. So here you can see the two compartments, acidic compartments stained nicely with eosin, and also the basophilic compartments of cerebellum where the granule cells are located. And this picture is from the skeletal muscles. These are the units of skeletal muscle fibers because they contain proteins, so they are nicely stained with eosin, an acidic dye. And in between these muscle fibers you can see these blue dots, which are actually the fibroblast cells formed in the connective tissue elements like collagen bundles that limit these muscle fiber bundles in the form what we call epimysium or perimysium, where you can see these elements fibroblast synthesizing these collagen molecules which are deposited extracellularly. So here also you can see the hematoxylin and eosin preparation, and you can see nicely that all muscle fibers are stained, and if you see any problem you can come to a conclusion. Like here you can see that some are very larger in profiles, but some are very small, some muscle fibers are very small. So that tells you something is going on with the skeletal muscle, maybe some degeneration. So this is how we can comment histopathologically about the status of a skeletal muscle. So we have to count the bulk of the fibers and their diameter, and fibers which are disproportionately smaller like this. So these are not sectional artifact, but this is due to some problems with the skeletal muscle fibers where degeneration might have started because of this loss of skeletal muscle proteins like actin or myosin. This is showing one low power view of pancreatic exocrine part. As you know, pancreas has exocrine and endocrine part. This is part of the exocrine part, which is composed of acinar cells. This is one magnified view of the acinus, pancreatic acinus, composed of acinar cells. And here you can see the cytoplasm contains lots of granular materials. These are actually protein secretory materials. Ideally, they should have been stained in pink color because these are acidophilic granules. So should have been stained in pink color. But as somebody asked me, so the reason is that these proteins may, cytoplasmic proteins may have both amino groups and carboxylic groups. So because of this differential reaction, we do not see an exact pink coloration of this acidic dye. Similarly, if you see this condition where the nuclei are also imparting a color which is not perfectly blue, that may be also due to this interaction with many proteins or nuclear proteins present in this kind of acinar cells. This is one skeletal muscle picture of skeletal muscle fibers. There are three fibers. These are extremely specialized cells, elongated cells. You can see elongated cells. Cytoplasm is differentiated in a elongated fashion, and where you can see the cytoplasmic proteins are organized in the form of bands, banded structures, cross striations, which are nothing but the disposition of actin and myosin along these cross striations. So this is one character that can be stained nicely even by eosin. And the components that are stained with hematoxylin, which is a basic dye, are the nuclei of these skeletal muscle cells that are located to the extreme periphery of these cells. So these are the nuclei located towards the or to the extreme periphery of these cells. So you must remember this is one unique feature for the skeletal muscles where the nuclei are shifted towards the periphery of this cell. This is one example of cartilage stained with hematoxylin and eosin. Eosin nicely stains these tissue materials, which are in fact the collagen fibers that forms the capsule of this cartilage connective tissue, a type of connective tissue, collagen fibrils. And then the main component of cartilage is the chondrocytes that are stained with the basic dye like hematoxylin, where you can see there is a gradient, means mature chondrocytes are located somewhere here and earliest chondrocytes are located towards the periphery of this cartilage. So this is how we describe the structure of a cartilage. This is one example of hyaline cartilage. Neurons can be stained with specialized techniques using special stain like thionin that stain nuclear substances present inside the neuronal cytoplasm. So these blue dots are nuclear substances that is stained with thionin. Sometimes we may need to see the entire neuronal profiles, and in that case we use Golgi staining, and with this we can see the entire profiles of neurons. Here you can see the cross section of a neuronal perikaryon where neither dendrites or nor axonal projections are clear, but here you can see the entire profile of neurons where you can see the cell body or perikaryon, the processes, some of which could be dendrites and some of them could be axonal projections. So from this shape and size and the number of projections, we can tell them what types of neurons are there in a particular region of the nervous tissue, whether they are motor neurons, multipolar neurons, fusiform neurons, pyramidal neurons, or horizontal neurons. This is from the cortex showing multipolar neurons stained with Golgi staining. And neurons also contain myelin in their myelinated axons, and this myelin can be stained with one special stain or dye, with one compound. This is neither a stain nor a dye, one chemical compound called osmium tetroxide. Osmium tetroxide, that is the best known lipid fixative and also lipid stain. So you remember, this is a lipid fixative as well as a lipid stain. Here osmium tetroxide is utilized to show the myelin lipids and in fact the myelinated axons present in a tract. So these are cross sections of the axons, and you can see the boundary where myelin is there, and myelin is essentially composed of lipids. Now in some cases we need to see some specialized structures by using histochemistry. Like in some tissues we need to see presence of carbohydrates and carbohydrate rich macromolecules like glycogen in liver and skeletal muscles, mucus in various cells like in gastrointestinal cells and respiratory epithelium, and in the epithelial basement membrane like in kidney. So we use one method called periodic acid Schiff reaction. There are two reagents, periodic acid and Schiff, and this actually helps in seeing the glycoconjugates present in the basement membrane of glomerulus. This is the glomerulus composed of afferent and efferent arterioles and lined with one basement membrane that is what we call pus positive, that is stained with pus. So glomerular basement membrane of kidney, and this simple staining method can tell you whether there is a problem of the glomerulus. Like in diabetic nephropathy, you can see that this glomerular basement membrane thickens exceptionally. This is in fact from a case of diabetic nephropathy showing a thickened basement membrane of the glomerulus. Otherwise this appears very thinner, like this kind of lining. These are the linings of proximal convoluted tubules of kidney. So from here we can tell that this sample is having some problems like pathological symptoms of diabetic nephropathy, etc. Now whatever we have said is just for our academic or knowledge based technique, means to see the histological organization of cells and tissues, their organizations in normal conditions, so that we understand the structures and correlate them with function. But in many cases there must have a clinical correlation. That means if we see some cellular structures, then definitely there must have some correlation and definitely clinical correlations. So here pathology is involved. So what happens in this kind of activities where clinical correlation is needed, where we need to correlate our observation with some clinical settings. So one pathologist from the pathology department may be asked to process a sample to see and diagnose the components of that tissue that is obtained at surgery, the tissue that is obtained in surgery, and then he will guide the surgeon to what extent the surgery will proceed, means whether this tissue is suffering from massive damage or neoplasm, what is the exact location of this neoplasm and where is the normal tissue located, so that the surgeons come to know all about this information of histopathology, and he will start dissecting the degenerative tissue in that patient. So what happens in when this kind of clinical correlation is needed? So the pathologist will immediately fix the tissue once it is received from the surgical department. So immediately he fixes the tissue in liquid nitrogen and he would cut in a cryotome that operates at a low temperature, say minus 22, minus 25 degrees Celsius, and after that he would stain with simple stain like eosin or hematoxylin, or if needed with some special stain, and he will come to know whether there is any neoplasm or to what extent the neoplasm has invaded the tissue, etc., and accordingly he can guide to the clinician for further resection or surgery. So this is the basis of use of frozen sections for having clinical correlations. So that is all about this lecture, and I would suggest you to read this categorically whenever you have time, because the entire chapter is contains about 170 pages, and it took about 1 month for me to read everything, to brush up my previous knowledge. So you may take some time, but please read categorically and read seriously so that you understand everything. Everything is understandable if you read categorically with full concentration, word by word. If you do not understand, so you again read, and you read repeatedly, then after some time you will definitely come to know what the meaning of that sentence is. In that case you have to exercise a lot, so you have to read cautiously about the cell structures, functions, etc., etc. Thank you.
Excuse me, sir.
Yes.
Sir,
Yes.
sir, I don't understand this frozen sections concept. Like, why is it used and how,
See,
like the procedure of it also.
See, normally, if we want to see only the histological organization of a tissue, say kidney or lung or liver or spleen, so we will process with normal method of histology, like we will fix in Bouin's fluid or formalin, and then we will go for paraffin embedding, and we will cut sectioning with a rotary microtome that operates at a room temperature. But
Yes, sir.
this is time dependent. This will take about 2 to 3 days. But when a surgeon needs to know during operation what is the state of neoplasm, how far it has progressed, so immediately he will forward this resected sample to the pathology department. So the pathologist would immediately cut by fixing the sample in liquid nitrogen, that is by freezing in liquid nitrogen. Then while in frozen he would cut the frozen sections in extreme low temperature, and then he would stain with suitable dyes in order to see what is the actual picture, so he can guide to the surgeon. So this is the use of frozen sections. So
Okay, sir, fine, sir.
okay.
I understood. Thank you so
Yeah,
much. Thank you, sir.
but in many cases we can also use also frozen sections, like when you attempt to see, say, localization of certain molecules in a biological tissue, we can also use frozen sections. There we can use special stain, or we can use antibodies to label those special molecules. Okay.
Okay, fine, sir.
Okay, thank you.
Fine, sir.
Thank
Thank
you.
you, sir.
Thank you. So I have given my mobile number and also email ID. You can also meet me, and I would also suggest that you must procure this book, which is very important, by Michael Ross, a must for everybody, and this is available from your seniors, and if not, you can procure even from me. I can give one electronic uh, mm, module purchased by me. Okay, so you must have this book. This is a must for your entire histology, cell biology, and many other, even for entire cell biology, protein synthesis, and molecular mechanisms of protein synthesis, transport, etc. You need this book. This is like the Bible of cell biology.
Can you just email us the activities
Uh,
and the practical work?
I don't know how you will get them. Either you have to come to me, or I can send by email. But this will be made available to. You will definitely get this PPT. This can serve as a guide. This can serve as a guide because in your uh, book you may find lots of information, and right at this moment you don't have to read everything, because you will read repeatedly in physiology and biochemistry classes. A number of lectures are allotted for this kind of activities like structure of organelles and their functions. Okay, so you don't bother for that. But you can collect this PPT from me, or I can send by email.
So can you upload this PPT on Serral platform?
That I will do. Okay. Okay. Nice.
Sir,
Yes.
sir,
Yes.
sir, is there any necessity like acidic proteins must be stained with basic dyes and basic proteins with acidic dyes? Is there any necessity as such?
No.
In general, proteins can be used.
It is. See, this is a very complicated issue, and it is not easy to delineate all this by simple staining under a light microscope. It is not easy. In that case, you have to employ a different staining method for acidic proteins and basic proteins. This hematoxylin and eosin may not be enough to discriminate one from the other. So there are methods. There are methods to stain acidic proteins or basic proteins. So hematoxylin eosin is a generalized staining methodology to see the proteins in general.
Fine, sir. Thank you,
Okay,
sir.
okay, thank you.
So,
Yes, yes.
is there something special about metachromatia?
Mm, Not really. It depends what you want to see. Like if you want to see glycosaminoglycans and all those present in the extracellular matrix, so maybe eosin and hematoxylin staining is not enough. So in that case, we can use metachromatic dye to especially locate all those chemical components. So this is the essence of metachromacia. Otherwise, we don't perform for general histology. So that is one. To see the entire distribution of mast cells in a connective tissue, okay, like in the peritoneum, or when you want to see or show the extracellular matrix in hardest elements like in cartilages, etc. So you need to perform this special staining using metachromatic dye. One example is toluidine blue, methyl blue, etc. Even they are limited.
Oh, thank you.
Okay, thank you. So if there are no questions, should we close down this lecture schedule? Sure? Sure.
Sir, I didn't understand the metachromasia concept totally. So please, you can repeat a bit.
See, metachromasia means certain specialized cells are rich in anionic components like sulfates and phosphates groups, huh, which are present in extracellular matrix like glycosaminoglycans, like heparan sulfate proteoglycans, chondroitin sulfate proteoglycans, which are present in extracellular matrix in specialized cells like cartilages. So they cannot be stained by simple routine eosin and hematoxylin staining method. In that case, we need to utilize metachromatic dye like toluidine blue that will clearly delineate the exact localization of those mucosubstances, glycosaminoglycans in those specialized cells. So this is what we call metachromasia that will afford exact localization of those substances by using one classic basic dye like toluidine blue. So this is metachromasia, and the color will be imparted in a different in different color. Staining will be imparted in different colors. Okay, maybe sulfate groups will be more red, whereas the phosphate groups will be somewhat purplish color. So thereby you can discriminate where heparan sulfate is located, where chondroitin sulfate, etc., etc.
Okay, so thank you, sir.
Thank you.
Sir,
Yes. Yes.
and sir, in if vesicles are transferred between the cis Golgi complex and the trans Golgi complex, even then the vesicles are coated with the COP type 2 protein.
Vesicles are put into.
If vesicles
No,
are
no.
transported from
Yes, yes, yes. See, one that leave the area, rough endoplasmic reticulum, and will move toward the cis Golgi phase. They will use COP2 protein in their coating, membrane coating. Whereas those that will travel from the trans-Golgi network to rough endoplasmic reticulum, where by mistake the proteins were transported, but these proteins were not either glycosylated or not correctly folded. So these misfolded proteins will be again sent back to the rough endoplasmic reticulum via COP1 coating. It is something like what is happening, what happens in a post office. Okay, if you don't use the correct pin code, it will be sent back to the sender. Sender is here rough endoplasmic reticulum, utilizing this COP1. And post office has different targets to send those return mail. Okay, via main post office.
yes,
So,
understood, sir.
okay, thank you.
Thanks.
Okay. So I leave this lecture class.
Regulatory secretory pathway.
Pardon? Yes,
Regulatory secretory pathway.
regulated.
Regulatory secretory pathway.
Okay, yes, there are many protein molecules that are synthesized and packaged and stored in the cytoplasm, but they are not released immediately. So, only when certain stimulus arrived at those cell, they will be triggered and they will be released extracellularly, like what happens in case of exocrine pancreas and in the neural structures when calcium enters in presence of cells need. Okay, then these vesicles will fuse with the calcium that has entered the cell, and they will migrate towards the cell periphery where they will fuse with the apical plasma membrane and will release their content. So this is called regulated. It is regulated by cells activities. Otherwise, the opposite is with the constitutive secretory mechanism, in which the vesicles once synthesized will be released immediately without storing. So you can find that these vesicles which are released extracellularly, those kind of cells under TM will not show any secretory vesicle stored in their cytoplasm because they are already released. But cells that store secretory vesicles and release only after certain needs, cells need, means which are regulated by cells activities. So in that case you will see that under TM those kind of cells will show the presence of secretory vesicles, those black dots in their cytoplasm. So this is the meaning of regulated secretory pathway of exocytosis. Okay.
So, sir, calcium will regulate it.
Yes, this is one example. There are many more stimuli, many more ligands which then bind and then trigger these uh, regulated activities. I am just telling about the action of calcium in the neurons and pancreatic acinar cells. Huh? There are hormones. Hormones when binds to these receptors, specific receptors, again those cells will be activated, and this activation will lead to the release of those secretory vesicles. So dependent upon the hormones as a stimulus or calciums as the messenger, so the cells will be activated. So that is the essence of this regulated secretory pathway of exocytosis. You will read a lot about hormone receptor signaling when you read the endocrine system. Okay. पहले तो म्यूट कर दीजिए मुझे, तो खत्म ही नहीं होगा। 12 मिनट एक्स्ट्रा हो गया तो अभी भी 56 है, कुछ नहीं कर सकते, क्या करें। इतना टाइम नहीं दे सकते।